Hi, my name is Shubham Anand and here's an abstract video on OptoGap, an optogenetics-enabled assay for quantification of cell-to-cell coupling in multicellular cardiac tissue. Putting in simple terms, how do you use optogenetic technology in cardiac tissue to measure electrical coupling between cells? First of all, let's look at the optogenetics and what is optogenetics and cell coupling. Optogenetics is a biological technique used to initiate or stimulate a response in a living cell tissue using light. Ion channels in cells are genetically engineered to react to light. For example, ion channels channel rhodopsin 2 programmed to react to blue light. This results in control of cellular activity such as opening and closing of ion-specific channels to make a cell excitable or inhibitory. Here is another important term known as intercellular coupling. Intercellular coupling means joining of two cells with gap junctions in order for action potentials to travel. The gap junctions also have connexin protein subunits. These subunits allow action potentials to travel very quickly, which makes intercellular coupling possible. How do optogenetics work? Wires carrying the opsin, for example channel rhodopsin, and identifier for specific cell type, which is usually known as a promoter, are injected into the tissue to infect the cells of interest. Wire spreads into the localized tissue as shown, and then lasers are used to activate these ion channels to change the activity of infected cells. What are the existing methods to assess intercellular coupling? The common way for measuring intercellular coupling is the dual cell patch clamp method. It measures the gap junctional current between two connected cells by using an electrode, as shown in the diagram, which contains an ion solution and amplifier to record minute voltage changes in cells. This method is strictly limited to isolated cell pairs with relatively high coupling resistance, which means it is not applicable with the native multicellular design of the experiment and reflects very low scalability ratio. Optogenetics is a better option for measuring intercellular coupling because it can provide cell specificity in a multicellular model. Also, it can be used in case of heterocellular electrical coupling, meaning coupling between different types of cells such as cardiomyocytes and non-cardiomyocytes. Let's look at the overview of this video abstract. Our aim is to measure multicellular electrical coupling of cardiac tissue. We hypothesize that using optogenetic actuators may help to measure electrical coupling as compared to existing techniques such as cell patch clamp method. Next, we will look at the two methods, 2D and 3D experimental designs, later on results and findings, and lastly discussion and conclusion. 2D experimental design. To demonstrate OptoGap, an in vitro 2D multicellular system was designed, consisting of a patterned region of cardiac fibroblasts expressing the light-sensitive actuators. The actuator used in this experimental was channel rhodopsin 2. A second layer of ventricular cardiomyocyte was placed on the top as shown in the image. Research studies show optimized generation of light-sensitive fibroblast channel rhodopsin 2 yield a consistent expression efficiency of more than 50%. In this model, cardiofibroblasts and cardiomyocytes tend to make gap junctions in the axial direction, not in the plane direction as illustrated in the image. We confirmed that channel rhodopsin 2 can trigger global activation in the non-confirmed cardiomyocytes. Furthermore, by observing optical actuation map shown in the figure, it was also concluded that higher expression efficiency of channel rhodopsin 2 produces image of the map with higher resolution. Basically, higher the efficiency, the more clear the optical actuation map will be, showing an effective flow of electric current. Validation of 2D experimental design. To validate the application of this method, other cell types were also tested. For example, coupling between human iPS-derived cardiac progenitor cells, which were made light-sensitive, and iPS cardiomyocytes. Why were human iPSCs-derived cardiomyocytes were used? Because they are a continuous source of cardiomyocytes from the same genetic background for use in multiple experiments. Therefore, these cells provide a major advantage over primary cells. Optical stimulation of the iPS cardiomyocytes through the channel rhodopsin expressing cardiac progenitor cells were successfully demonstrated in this heterocellular model shown in the figure. Now we look at the 3D experimental design. The experimental validation of OptoGap encouraged computational analysis to further understand the limitations of the method, especially when applied in the complex 3D heart setting. Using the cell pair model shown in the figure on the right, where the cardiofibroblast is optogenetically changed, light stimulations with relative low, medium, and high were performed with gap junctional coupling strength of 2 and 5. Cell delivery of channel rhodopsin clusters were stimulated and localized with the native non-optogenetically modified cardiomyocytes at the left ventricular apex as shown in the diagram. These clusters covered a range of cell densities from 0.05, 0.1, and 0.25. Results showed that within each density, successful heterocellular coupling were reported despite very different spatial patterns of clustering. Discussion. There are still several challenges in fully developing optogenic methods, including optogap, in the whole heart. Light penetration is limited into the dense muscle tissue, especially in the case of blue light. This problem can be partially solved by employing red-shifted options such as REACH R45 or Crimson R46, which would permit engaging cells from deeper layers. Furthermore, results show that both the use of optogenetic sensor or optogenetic actuators can help in detecting heterocellular coupling. However, there is a difference in the response, such as the action of the optogenetic sensor in non-cardiomyocytes usually leads binary outcome, absence or presence of, which means absence or presence of a signal. Why channelrhodopsin 2 was used in the experiment? In this experimental study, channelrhodopsin 2 is an ion channel. When stimulated with a blue light, allows positive ions to enter the cell. When positive ions enter the cell, membrane voltage increases positively above the cell's threshold. This increase in voltage starts an action potential. Then these APs travel to the neighboring cells via gap junctions to carry out the electrical coupling. Because the main goal was to measure electrical coupling in cardiac cell tissue, channelrhodopsin 2 plays an important role in this experiment. Conclusion. Throughout this experiment, scalability and automation were the aspects challenged and need to be considered for better understanding in cardiac electrophysiology. OptoGap was able to provide a readout of the electrical coupling from complex 3D multicellular design. In general, optogenetic methods were well suited for probing cellular intercellular interactions where cell specificity is needed. Credits. Thank you for watching.